Welcome my dear students to this section. Now we'll be taking the nucleic acid chemistry further. We saw how purines were synthesized, how purines were degraded, how pyrimidines were synthesized, how they were degraded. Now there are in various textbooks, they give it at different points in the textbook. I have included them in the molecular biology section because I feel ATP synthesis or any of the purine synthesis and degradation is all dependent on related to molecular biology and the structure of DNA. So let us go ahead with the structure of DNA. Now, who gave this structure? A bit of history. I'll be talking about this paper was the structure of DNA, as we all know it, was given by Watson and Crick in 1953. The molecular structure of nucleic acids, a structure for deoxyribose nucleic acid, was published in Nature, the journal Nature, 171, 737 to 738 in April 1953. And in 1962, the Nobel Prize was given to these three people, Watson, Crick and Wilkins, for their contribution to physiology. Nobel Prize in Physiology or Medicine went to Watson and Crick. There is a lot of story behind how they went about, how the race was going on, how Rosalind Franklin and all of them helped Watson and Crick to come out with the structure of DNA. So let us see what this structure is, which has created so much, which is necessary so much for our body. Let's say now DNA is basically deoxyribonucleotide is deoxyribonucleic acid. It is a polynucleotide made up of deoxyribonucleic acid. The name is deoxyribonucleic acid. It is a polynucleotide made up of deoxyadenylate, deoxyguanylate, deoxycytidylate and deoxythymidylate. Just for the sake of this, you will have to know the names. Otherwise, you can know it as DAMP, DGMP, DCMP or DAMP. Now the nucleotides are attached to each other by the 3', 5' phosphodiester bonds to form one long polynucleotide strand. There are two such polynucleotide strands in a DNA. Each strand will have a free 5' phosphate group and a 3' hydroxyl group. So here I have shown you that how the nucleotides are attached to each other. Now the sugar and the phosphate is going to form a backbone and the bases are going to be inside this backbone, inside the chain. So it has a 5' phosphate group at the 5' position and at the 3' position it is going to have a hydroxyl group. Why do I say 3' and 5'? Please remember there are carbon atoms and nitrogen atoms present in the base also. The base may be in any sequence. Whatever is the sequence is important, and it is the sequence which is responsible for the function of the DNA or for a different gene. Everything will depend upon the sequence of the base, and this is known as the base sequence. So, but whatever it is, all the DNA molecules, whichever strand of DNA you take, it is double stranded. Now what I have shown here is a single strand of DNA. Double strand, there will be two strands like this. One strand will be made like this with each of them will be having a 5' phosphate and a 3' hydroxyl group. So if you look at it, actually this DNA is always having a 5' end and a 3' end. So it is having a polarity. At the 5' end always will have a phosphate group. At the 3' end it will always have a hydroxyl group. So this polarity, and plus we say that each strand is running anti-parallelly. When I say anti-parallel strand, what do I mean? The other strand will be from this direction to this direction, anti-parallel. The strands are anti-parallel. So because of this presence of this phosphate group and because of the presence of the hydroxyl group, it gives it a direction. A direction is given to the DNA. By convention, we always read from the 5' to the 3' end. I'll talk more about this as we go ahead. So let us look at and remember this strand, it is made up of the sugar phosphate backbone and the bases are going inside. Now in a double strand, the base of this and base of the second strand, they are going to form hydrogen bonds, and this is what is going to stabilize the DNA structure. So let us talk more about this. We'll go further. So what is the importance of this nucleotide sequence? As I said, the sequence of the base is important. It forms the unique genetic information of an organism. This organism will have this nucleotide sequence. That is, the sequence is the uniqueness for that organism is because of the nucleotide sequence. Now, if there are two organisms, if they are supposed to be related, then the sequences are more related. So, closer the relationship between two organisms, more similar the DNA nucleotide sequences will be. As far as evolution is concerned, if you are going to say from different organisms, different things have evolution has taken place. So if you want to know the primate which is most closer to the human being, then the DNA sequence between that primate and the human being will be more closer. The nucleotide sequences will be more similar. So that is the reason why we can look at the DNA sequences to know the evolutionary pattern. Now, scientists use this nucleotide sequence to determine evolutionary relationship among organisms, to tell from which organism has evolved, to determine whether two people are related. I think we all know about forensic science, and to determine whether two people are related, whether there is a paternity dispute, and we want to say that that person is going to be the son or the daughter of a particular father, then we can do a DNA sequencing. Because they are related, we can able to know the nucleotide sequence. If there is a particular way it is there, then they are related. We are talking about paternity disputes here, or to identify bodies of crime victims. We'll talk more about this, the application of molecular biology, but everywhere it is the nucleotide sequence that is important. It is the sequence which is going to determine, which is used for all these purposes. Now let us go further with the structure of the DNA double helix. Now Watson and Crick, they propose that the DNA is shaped like a long zipper, and that is twisted into a coil like spring. So there are different ways you can show this DNA double helix. So for the sake of convenience, I'm going to show it like that. So you have got this zipper, and this zipper is folded among, it is going to form a double helix. So the double helix, double helix is being formed by this zipper. So you have this different base sequence like a ladder, and this ladder is forming a double helix. So you have the bases which are going to form and stabilize this DNA structure. Okay, this is how, and remember, each strand is having its own polarity and it is directional, and so you will have, let us say, 5 prime. So this will be your 3 prime end, and this will be your 5 prime end. So this will have a double helical structure. Now, because the DNA is composed of 2 strands which are twisted together, this shape is called as the double helix, and it is a right-handed double helix. Right-handed double helix. So in this double helix, as I said, the bases are going to pair with each other. We have got to what is called as Watson-Crick base pairing rule. So let us look at this base pairing rule. The 2 strands are complementary to each other, and base pairing means always adenine is going to pair with thymine, and it is G with C. Now I have shown here, what is this? What are these? These are hydrogen bonds between these bases. A always hydrogen bonds with T, G always hydrogen bonds with C. This is the Watson-Crick base pairing rules. The number of bonds between A and T is always 2, and between G and C it is always 3. How do you remember this? T stands for 2, so AT it is always 2. So the 2 strands are complementary to each other always. So if you have A If you have A here, it will be T here, and depending on the number of G, C, C, G, they are complementary to each other thanks to this base pairing rule. So that is the meaning of complementary. And plus, along with this, we have one more scientist who has come out with this rule, which was responsible for Watson and Crick coming out with a double helical structure and this Watson-Crick base pairing rule. Now, what does the Chargas rule say? Again, MCQ point of view, this is necessary. The Chargas rule says that the amount of A is always equal to amount of T, and then G is equal to C. Because they are complementary base pairing, naturally how much ever adenine is there, that much thymine is going to be there. How much ever guanine is there, that much amount of cytosine is also going to be there. So they are, that is one part of his rule. The other part, he says that the amount of purines is equal to the amount of pyrimidines. That is, A plus G is equal to C plus T. So in any given double-stranded DNA, if whatever be the structure, amount of A, if there are 20 adenine nucleotides in a given DNA strand, then number of thymine will be 20. If there are 30 guanine, then the number of cytosine will be 30. At the same time, this has to give one more feature, that is A plus G, that is 20 plus 30 must be equal to 20 plus 30. That is, A number of purines must be equal to the number of pyrimidines. So this is the meaning of the Chargas rule. This Chargas rule is used even today. Suppose you have got a single strand of DNA, you will want to know the sequence of the double strand of the DNA. We use the Chargas rule. The DNA strands, let us go further, are held together by hydrogen bonds. There are 2 between A and T and 3 between G and C. Now the 2 strands run anti-parallel to each other. One of the strands is known as the sense strand and the other strand is known as the antisense strand. What do you mean by that? The strand which contains the gene and which is going to act as a template for during transcription. Based on this, we will have the sense strand and the antisense strand. I'll talk about transcription more as we go. So remember that each of these strands, depending upon the position of the gene, can act either as a sense or an antisense strand. So this is the basic sense and antisense strand. These are terminologies that you should be aware of. Now let us go further. Now in a DNA, when it is forming a double helix like this, it will result in the formation of what is called as the major groove and the minor groove. Now in my diagram, it may not be so evident, but usually there will be a major groove and a minor groove. Minor groove is smaller in size and a major groove is bigger in size. Major, bigger in size, and a minor groove is smaller in size. So what is the function of these grooves? Let us look at this. Now the minor groove exposes the edge from which the C1 or prime atoms extend, and the major exposes opposite edge of the base pairs. That is just the way, because of the helical structure, this is happening. Now what is important is both major and minor grooves are places where proteins interact with DNA. So whenever a regulatory protein is going to regulate gene expression, it will be at the level of the major groove or at the minor groove. There are particular protein motifs, what we call as zinc motif, DNA zinc finger. All these motifs are responsible for regulating the DNA, regulating the gene expression. So when it is going to regulate, it will need a place whereby it can come and bind. Suppose a protein is going to have a region to go and bind, then it requires a groove. So hence we have got major groove and minor groove. Main function is those are the places where proteins interact with the DNA. So now I'll go ahead with the different forms of DNA. Which are the different forms of DNA? Now there are 3 major forms: bDNA, aDNA and zDNA. zDNA, the word has come because it is zigzag, because of the way those base pairs are arranged. Out of all this, bDNA is the major DNA. bDNA is biologically the most common. It is right-handed and having a 20 Armstrong diameter. So again, We are looking at the diameter MCQ point of view. So just remember that it is right-handed and has having a 20 Armstrong diameter. It has complementary base pairing, that is Watson-Crick base pairing of AT and GC. If you look at the geometry of bDNA, ideal bDNA has got 10 base pairs per turn. The pitch is 10 point into 3.4, 34 Armstrongs per complete turn. The axis passes through the middle of each base pair. The minor groove is narrow, major groove is wide, and that is a simple characteristics of bDNA, the most common DNA. What about aDNA? Again, this is also a right-handed helix. I'm just talking about this from point of view of MCQ, where it might be asked. It is right-handed helix. This is wider and flatter than bDNA. It has 11.6 base pairs per turn and a pitch of 34. The base planes are tilted at 20 degrees with respect to the helical axis. The helix axis passes above the major groove, and deep major and shallow minor grooves are present in aDNA. Usually it is observed under dehydrating conditions. zDNA is a left-handed helix. As I said, it stands for zigzag. Seen in high salt conditions, it has 12 Watson-Crick base pairs per turn, a deep minor groove, no discernible major groove, and reversible change from bDNA to zDNA in localized regions may act as a switch to regulate gene expression. Maybe it is involved in the transient formation. Maybe it is zDNA is transiently formed whenever there is a actively transcribing RNA polymerase. Maybe that is the reason it is transient formation may be there. But more than that, zDNA is usually seen in high salt conditions. MCQ point of view, hence I am telling about these. Now let us go to RNA. We finished DNA, let us go to RNA, ribonucleic acid. So it is single-stranded. Compared to DNA, this is single-stranded. But there will be areas in certain RNAs can be double-stranded, sort of, in certain conditions. But let us stick to this. RNA is single-stranded. The sugar is ribose, contains uracil. Chargas rules are not obeyed. That is, A plus T is not equal to the T, or G is not equal to C, or A plus G is not equal to A plus the amount of purines is not equal to the amount of pyrimidines. Which are the different types of RNA? Messenger RNA, transfer RNA, ribosomal RNA, snRNA, hnRNA. One more is there, I'll talk about this. So mRNA, tRNA, rRNA and snRNA or small nuclear RNA. So let us look at hnRNA or heterogeneous RNA and mRNA. I'm just giving a very brief overview about this. I'll talk more about this when I'm talking about transcription. So hnRNA is transcribed from the DNA. So when the DNA undergoes transcription, it forms a long single-stranded RNA molecule. This is known as the hnRNA. hnRNA is having a lot of what we call as exons and introns. Now it undergoes processing and a process called as splicing, and then hnRNA is converted to mRNA. Now mRNA is characterized by certain characteristic features. It has a 5' end capped by a 7-methyl guanosine. So as I was telling, at the 3' end it has got a poly A tail. So 5' end mRNA will always have certain characteristic feature. Let me just write it to you. mRNA is single-stranded. It is got from the DNA, and mRNA has a characteristic feature at the 5' end. It has got a 7-methyl guanosine. Guanosine is present at the 5' end. At the 3' end, it has a lot of A, and this is called as the poly A tail. So it is a head and a tail, you can say. This is at the 3' end, and this is mRNA. Now each mRNA can be translated into a polypeptide. let us look at the next RNA, that is transfer RNA. Transfer RNA is around 75 to 90 nucleotides. They serve as adapter molecules during protein synthesis. There are 20 different transfer RNAs corresponding to each amino acid. The structure is described as a clover leaf model with 4 arms and a stem. So I will be talking more about this as we go, when I am talking about transcription. So for now, remember that it has got 4 arms and it is a clover leaf structure. Now the 4 arms, it has a variable arm, an anticodon arm, amino acid accepting arm, T-cyc arm, dihydrouracil arm. I'll talk about more about this when I'm talking about transcription. This is a tRNA molecule. This is a tRNA molecule. I'll talk more about it as we go ahead. Now, given certain characteristics here, the acceptor arm, the anticodon arm, the D arm, the T-cyc arm which has pseudouridine, and the variable arm. I'll talk about all these functions when I'm talking about transcription. Now another RNA is ribosomal RNA, rRNA. rRNA is present along with ribosomes. So protein plus RNA becomes a ribosome. So the 60S subunit has got these rRNAs, 28S, 5S, 5.8 and 40S has got 18S RNA. What are these S? Everything we'll talk about it in transcription. Just keep a note of this. Small nuclear RNA, where are they present? Small nuclear RNA are present in the nucleus, hence the name snRNA, and their main function is in splicing. So let us talk about the different RNAs. I'll talk about them again when I am talking about transcription. So for right now, just remember that ribosomal RNA is around 28S rRNA, 5S rRNA, 5.8, 18S, etc. So with this, we come to the end of structure of DNA and structure of RNA. So let us finish the nucleic acid structures. We'll talk about this. Talk about what are replication, transcription, translation as we go ahead. Thank you.